This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode 209, recorded on November 30th, 2012. Hi, everyone. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. This episode of TWiV is sponsored by ASM, the American Society for Microbiology, the world's largest membership society for microbiologists. Find out how ASM membership can help advance your science, your career, and your network. Go to asm.org/advance. Joining me today from here in the TWiV studio, Dickson Despommier.
Hello, Vincent.
I see you twice in 1 week.
It's amazing, isn't it?
2 podcasts.
Hey, it's great.
I'm not going to ask you how you've been because I know already.
Exactly.
Although maybe those who listen don't know. How have you been?
I've been fine.
You've
thanks.
been in Italy, right?
I have, yes. And it was a marvelous experience.
I have to agree with you.
Why? I wonder why, Vince.
Yeah, let's think about that.
Let's check his last name, everybody. Let's find out.
First generation.
Aha! That's
Right.
important. That's very important.
Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody.
How's Ann Arbor?
It's fine. We have, where to go? There, 45 degrees and a sunny day, and that's 7 Celsius.
Is football still on out there, or is it finished?
It's finished.
Thank God.
Finished.
It's done, huh?
Yeah.
There are no bowl games or anything?
Oh, I haven't kept track of whether Michigan will go to a bowl game. I think they will, but not to a BCS bowl. I don't know.
Are you a football fan?
Yes, but more for my alma mater, Purdue.
I see.
Yes.
Do they have a good team?
No. Well, I mean, about 50-50. I think they won 6 and lost 6, but I think that might mean they can go to a bowl game, too.
Probably does. Kathy, do you know where I went to school?
No.
Went to Notre Dame.
Oh, you are the enemy.
We are the enemy.
And there you
And we're going to go to a bowl game.
Yes, you are. Definitely.
Also joining us today from Western Massachusetts, where there are no bowl games, Alan Dove.
Yes, indeed. Good to be here.
And there's nothing out there, right? There's
Nothing. There's just it's me.
0.
It's just me.
That's right.
That's the best situation. How have you been, Alan?
Oh, things are going okay. It's kind of dreary here now. Overcast, 1 Celsius, and supposed to get wintry mix this weekend, but that's the way it is here in December.
Here it is cloudy also, right, Dickson?
It is.
You're just outside. How's it feel?
It wasn't as bad today as it was yesterday. Yesterday was kind of raw with a little chill in the wind. Today it's tolerable.
It's 8 degrees Celsius at the moment.
Yeah, it wasn't bad.
Alan, you said it was 1 Celsius?
1 Celsius, 34 Fahrenheit.
When he was saying that, I heard him say 1 cell, and I stopped there. Said,
Yes, it's 1 cell up here, and I'm in it.
Protozoan-like weather.
They let me out every couple of weeks.
Are you parasitic?
I'm not sure.
Oh,
I'm not sure I would know.
Oh, I see. Rich Condit is due to join us soon. He is taking care of his plumbing and actual pipes in the house.
That's not metaphorical.
No, not at all. Not at all. All right. We have today's an all-email episode. And before we get to that, we have some follow-ups from TWiV 208, which was my conversation with John Udell. And the first is from Raul Andino, a virologist at UCSF. He writes, I was driving from Palo Alto to UCSF and listening to my favorite show, TWiV. Well, after the Soccer South American show, which is my real favorite. And I heard the discussion on changing the scientific system, making some experiment to try to save science in this country. So I want to say that you can count me in for this. I think that it is incredibly important. Probably we can recruit additional people here at UCSF whom are also very concerned about it. Let me know how we can help. Perhaps it would be a good idea to start an email discussion followed by an informal meeting, collecting ideas from people to enhance the proposal. I am naive on this, which is good and bad, and I imagine there are many strong forces that may not want to see big changes on the system, but a small-scale experiment may work. I like the idea of retrospective funding. Very nice programs, thanks. I even listen when I'm on my bike riding the Santa Cruz Mountains. Cool. Nerd. We just got a text from Rich Condit.
Yes, he's apparently getting hammered.
Really?
I mean, they're bathroom tile.
Hammering.
Yeah.
This could get ugly.
That's right. At least he's not feeling grouty.
Ooh. You don't look like Alan.
Alan, I can't do you. I'm sorry. I try, but it doesn't work.
The next one's from Robin.
Wait, let's go back to Raoul's a minute.
Yeah, sure.
Because I also was very inspired, and so I emailed John Udell. I think he has a lot of good ideas, and if there's things we can do to help, I'm in. So.
Yeah, if you haven't listened to 208, do so. He has some good ideas about how to fix the system.
In fact, it was my first thought when I had to come up with a pick of the week, but I thought that would be too self-referential to pick last week's TWiV. But I really think it's outstanding and worth listening to.
Okay. I'm glad Raul's listening. Hope he listens today because there's an email about an insect virus that he has to weigh in on. Mm-hmm. The next one's from Robin. His email was entitled Overlooking the Gun. John Udell needs to be informed that the government has nothing of its own. Everything it has been taken from others under the threat of coercive violence to enforce compliance. Anytime one talks about the government, one must keep one's focus on the gun of enforcement.
Mm-hmm. Unless you're writing from Syria, I think that's a tad melodramatic.
You think so?
And I know you're not writing from Syria because you can't email from there.
That's true.
Robin's here in the U.S. Probably a bit much, don't you think?
Yeah, the enforcement portions of the government, actually, I think of them as working for me rather than against me, because the purpose of enforcement for things like the tax code, which is really what he's going on about here, is to keep people from being freeloaders.
Yeah.
Mm-hmm.
So, well, you need money to do things, don't you?
Yeah, you need money to do things. And look, democracy's not perfect, but nobody in 10,000 years has come up with a better system.
This is true.
So what we do in a democracy is we have this organization that we refer to as a government, and we, the people, get together and we tell the government what to do, and the majority vote determines what the government does, and then we chip in and allow the government to provide all these wonderful services like the fire department and the police and roads and all that sort of thing. And science is part of that because this has been the economic engine for the development of what we now call the developed world for the past century. And yes, we all chip into that system and we all benefit from it. And that's something that I think some old dead guy long ago said was the social contract.
Right.
The social contract is a little like one of those shrink wrap agreements on software. You don't actually know what you've agreed to until you're already in it.
That's right.
But again, nobody's come up with a better solution to that. So you've already signed on to this, and here we are. And if you don't agree with the majority, well, try and persuade the majority to agree with you. Until then,
yeah. John was very supportive. He said he was proud to work for the government.
Sure.
And he thinks they do good things. And he did say they work for us.
They do.
So.
Everybody I have met who works in a government post has been has been very conscious of the fact that they are working for the people and that they've been entrusted with with resources that they need to manage.
Yeah. The next one is from Chris Suspect, who writes, I am really enjoying the discussion with John Udell. At points, he reminds me of a right-wing political talk show host, not in the content or message, obviously, but more in his verbal delivery. He'd make a great host for a show dedicated to science policy. He has a great delivery. He's wonderful.
That's a good idea. TWISP.
TWISP.
Excellent. You'd probably like that yourself, Alan, right?
Yeah, I'd be into that.
I'll run the boards.
Okay.
We have had a bunch of transcripts sent in recently, a whole bunch by listener Steve.
which is awesome.
It's great. He's done one on Mark Chalberg, the one out in Nebraska, Huskers Go Viral, the TWiV special on XMRV. He even did the one on how to read a science paper. I haven't uploaded that one yet. And so he's on a roll. He wants to do a lot more. He's got some sort of system, almost mechanized, for doing it.
Well, thank you very much, Steve.
Absolutely.
Yes.
So that's great.
That's outstanding.
I also received one yesterday from John, and I'll read his letter. Attached is the transcript for TWiV 131. This was a rheostat for cancer, our discussion with Brad Thompson of Oncolytics Biotech. I did not include the concluding section where you answer emails on the air, but I finished the entire interview. Thanks for all the podcasts. I listened to them all. John is a postdoc in mathematical physics.
Lord.
P.S. I have followed Oncolytic Biotech very closely since I listened to TWiV 131 about a year ago, and it only looks more promising today. Since that episode appeared, the NCI of Canada has announced funding for 4 roughly 100-patient randomized trials of real lysine in 4 different types of cancer. The NCI in the U.S. is also funding 2 similarly sized randomized trials. The Phase 3 interim data was expected out around early August 2012, but the trial design was changed. This change, which was announced in September, was made because the subset of patients with distal metastatic tumors but without local tumors were doing much better than the complement subset and also much better than expected given similar patient groups in studies for other drugs. The subset with local tumors plus or minus METs were also doing better than expected, but not as much so as the METs-only group, METs being metastases. Note that the data is still blinded, but the combined treatment plus control group was doing much better than expected. I view this as a positive sign, since you may recall that on your episode, Brad said that real lysine was more effective on metastatic tumors than primaries. Well, thanks for the update, John, and thanks for the transcription.
Yeah, and you know, I just thought of something. If I don't know. John obviously is just sort of doing this in his part-time between mathematical physics analyses, which I would find mind-boggling. But Steve, if Steve is doing transcripts professionally, could we insert an ad for him if that would help him?
I don't think so. I think he does something else.
Okay.
But he's in constant correspondence with me. He actually wants to set up a section of the TWiV website where it's dedicated to transcripts and telling people how to do it and all this. So
cool.
He's really into it.
Well, it just occurred to me, if we could set up some kind of quid pro quo here.
Yeah, sure.
We can plug somebody's services if they're transcribing.
Yeah, so if Steve has something we can plug, let us know. Yeah.
Yeah, I just counted up because you do have a page of the transcripts. There's 18 there, which is about 9%, counting today's episode.
Not bad.
Or so. Yeah. So we have a ways to go, but it's nice to have some of those the historical ones, and it's great.
Dickson, the first episode is there as a transcript.
Wow.
West Nile virus.
Cool.
Remember that?
I sure do. No, I remember the day you asked me to do this. And I said...
ran away.
I did, but you run faster than I do.
All right. Now for the email. Alan, can you start with Combs?
Sure. Colm writes, Twivsters, I'm writing to address a few of the points you've touched on recently. First, with regard to journal clubs and how to keep people interested in coming. Here at UAB, we're required to take a journal club every semester, most of which meet weekly. Mine in particular, University of Alabama, Birmingham?
Yeah. Yep.
Okay. Mine in particular uses a setup where each student gives 3 types of presentations. First, the traditional explain this paper, figure by figure with background, critique, and Q&A. Second, a discussion format where the assigned student puts together slides with the figures and a brief introduction that leads a group discussion focusing on the overarching biological question posed by the paper, whether it was adequately addressed and future aims. The third is a more casual, bring in any article, newspaper clipping, MMWR report, etc. you find interesting and say why in about 7 to 10 minutes. This is ideal for bringing up topics not suitable for our more molecular virology-focused journal club, but still of interest to an individual or the group as a whole. I think regularly presenting these different types of public speaking is a benefit, as one helps giving seminars, one is more teaching experience, and the other helps you communicate science in a casual way. We also recently began presenting series of papers on single topics, animal models, short RNAs, etc., to avoid jumping around vastly different fields every single week. I very much enjoy the way we have our journal club set up. Both of our faculty moderators listen to TWiV, but I swear I'm not just writing in to suck up. Recently, you fielded a question about, not that there be anything wrong with that.
No.
Fielded a question about trying to get TWiV onto NPR and the attendant difficulty. I think a more feasible approach would be going directly to what remains of college radio. Many of these stations, WMUC at my alma mater, Maryland, for example, are in some way overseen or advised by journalism programs and air some form of educational or news programming in between student-driven free-form programming. And since many stations have lost their FM licenses and moved completely streaming, it wouldn't even have to broadcast. WKCR could have an entire Twix substream. Another option would be reaching out to community-based low-power FM stations, either in developed or developing countries. But I'm not as familiar with them. Maybe Alan would be that resource. Hope I gave you some novel ideas. Cheers.
Is there a radio station at Columbia, Dickson?
There is. I'm trying to think of the name of it. WCKR.
Maybe it's KCR, like he said.
WKCR.
Yeah, it must be KCR.
I think it is WKCR.
89.9 FM.
That's right. That's right.
I occasionally listen to that. I more often listen to the Fordham one.
Yep.
So could I provide them with my stream on Fridays and they could stream it out?
Why not?
So, I mean, in theory, if they
Sure.
have
time, right? Yeah.
Yep.
If they have a time slot, but with the streaming options available now, they probably could put it somewhere.
Yeah.
Yep.
It's a good idea, I think, and we'll look into that. As many places as we can get.
Yeah.
I also like the journal ideas. Those are good.
Mm-hmm.
Kathy, can you take the next one?
Sure. Maria writes, "Hello, TWiV. First, I must admit that I have not been the most faithful TWiV listener.
That's it. Stop right there. Get it.
Next.
But I was amused to hear from my boss, Dr. Matthew Evans, that on TWiV, one listener wrote in asking about how best to stay mentally sharp and ready for graduate school while training full-time for the Olympics. And that in response to this listener, the TWiV crew stated that it would not be possible to simultaneously train at the Olympic level while earning a doctorate. Matt wrote in a response to TWiV, but I also felt compelled to enlighten everyone about being the ultimate student athlete after listening to the podcast myself. I am a PhD candidate at Mount Sinai School of Medicine, where I am studying hepatitis C virus entry in the lab of Dr. Matthew Evans. I just returned this September from London, where I competed in the 2012 Olympic Games representing our country, the United States of America. I am currently beginning my 5th year here at MSSM, and aside from a 6-month leave of absence spanning this past March to August, during this period of extra free time, I finally got to read Vertical Farm, great read.
Ho!
I have been a full-time PhD student and part-time elite athlete. I have raced at the international level since high school and have completed all over the world at various competitions, most recently London for the Olympic Games. I am a race walker, RW, which for anyone who has never heard of this, it is a discipline in track and field with the Olympic distance covering 20 kilometers. It is an endurance event often contested on the roads where the athletes race around a 2-kilometer or 2.5-kilometer circuit. There are 2 rules that differentiate race walking from running. The first is you must maintain contact with the ground. Therefore, there is a double contact stage as opposed to running where there is a mid-flight stage. The second rule is that the front leg must straighten on contact as it becomes weight-bearing and passes underneath the body, as opposed to running where the knee buckles and the quadriceps flex to catch one's weight. I was never homeschooled. I graduated from high school on time and graduated from undergrad a semester early. At graduate school, I've completed all my coursework on time and have even published my own first author paper. I did this while simultaneously earning and defending my indoor and outdoor nationals titles, in which I have currently racked up 3 indoor 3,000-meter race walk and 3 outdoor 20K 20,000-meter race walk titles. Last year, I finished 30th at Worlds, and this year, 29th at the Olympic Games. I spend on average 2 to 3 hours a day training and cover 70 miles a week while working a 10 to 12-hour day in the lab 5 days a week with light work over the weekend. I found it fitting how the next letter comment that was addressed on the same episode was regarding multitasking. In regard to those comments, I couldn't agree more. Yes, being a student athlete requires a lot of pre-planning and careful time management. I can definitely attest to multitasking during periods of incubations. For example, I may begin a transfer overnight, then the next morning head into lab, remove my blots, and allow them to block while I go outside and complete training for 1 to 2 hours. Then before showering, I return to lab again, add my primary antibody, and then while it's incubating, wash up and eat breakfast.
Wow.
Nearly everyone in my department has seen me come into lab on multiple occasions wearing spandex tights and my water bottle belt for training. I am a huge advantage since MSSM is located right next to Central Park, but I would be lying if I didn't admit that the school's proximity to a place for training was one of the reasons I chose it. I also agree that multitasking in the sense of playing on one's iPhone, surfing the web, or checking emails is unproductive multitasking.
Yeah.
That is why there has been little time in my life for these activities. In addition to my multitasking, planning, and jam-packed busy days, another huge factor contributing to my success was an understanding boss, Matt, supportive graduate school program, and an equally understanding and accommodating coach, Tim Seaman. There were plenty of times when I prioritized school over training, like when I was studying for my qualifying exam. The reverse is also true when I would use my vacation time and attend a pre-training camp in Daegu, South Korea before the Worlds, requiring me to be absent from lab for 10 days straight. Matt could have very easily forbade me from taking the time off that I did to compete, which was always more than the allotted 2 weeks per year a graduate student is entitled to. Together, everyone allowed me to reach my Olympic dream, and I was never forced to give up on my academic pursuits. Yes, this is often not the case. I would estimate that at least 90% of my Olympic teammates were full-time professional athletes, and the other 10%, I do not know another single person simultaneously earning a PhD.
Right.
While rare, it is not impossible. I think this would be a great comment for you guys to address. After all, a good number of listeners are probably undergraduates, and it is important for them to realize that they do not have to choose a less demanding career path if they want to stay active in sports. It is possible to train at an elite level and pursue your academic studies. Thanks for taking the time to read my letter. For more info about my recent Olympic journey, visit my website. She gives the URL, or check out the attachment to see what an Olympic PhD candidate looks like. I look forward to listening regularly again to both TWiV and TWIM.
Fantastic.
Cool.
I must also say that we had an Olympic champion in our medical school class. Her name was Jenny Thompson. She was a swimmer. She probably still is a swimmer. She had something like 10 or 12 gold medals, and she competed in the last Olympics also, so she was in medical school while that happened.
Wow.
Cool.
Yeah.
Yeah, I was...
And she graduated on time. She graduated on time.
I think I remember seeing her
She was amazing.
competing.
Yeah. She was in the
She's a
relays.
mother, I think, right now.
The Americans came in second in the relays, and she was one of the anchors for that team. And she won a silver medal.
Cool.
Yep.
So it can be done, and for a PhD,
It is being done.
For a PhD, it seems like the key is go to Matt's lab.
That's it.
Yeah, that's
That's
right.
it.
Well, I agree. I think this is fabulous that you can do both, but clearly you are very disciplined and focused.
And multi-talented.
Maria,
as well
multi-talented, and
That's right.
not everyone has those talents. So,
Yeah.
if someone is not as focused as you, it may not work. But I think it's a great example for people who want to try to do it.
Yeah.
Her website is great. There are wonderful photos of her hanging out with Obama and Michael Phelps and running in London. It's pretty cool.
Yeah.
So check it out. We'll have that link. I think she sent a picture of a postcard that she had sent the lab from London. So I will stick that up on the website as well. All right. So all you Olympians, that should be encouragement.
Or would-be Olympians.
Would-be Olympians.
I have a student who trained for the marathon this year.
Right. That's too bad because they didn't run it.
Yeah, they canceled it.
That was they must have had so many disappointed people.
So I don't say anything about her running all the time, so it's fine with me. I think even John Udell said last week it's good to get some exercise if you're working in a lab. Keeps your head clear.
Absolutely.
That's
When I was in graduate school, I used to do a lot of biking.
Yeah, I know.
Yes. Actually, yeah, you hauled my bike to a meeting once.
I did.
Yeah, it was a Gordon conference, and I
really?
rode up with you, and we put my bike on the roof rack so I could
cool.
do some morning rides while we were there.
I'm glad I was understanding back
Yes,
then.
you were. Yeah, no, I was not. I was certainly not at Olympic level or.
Well, there's still time.
There is still time,
right.
that's
Right.
Stay away from the doping. Hey, Dickson, could
Yes,
you
sir.
read Zachary's letter?
I'd be pleased to. Zachary writes, after watching your...
I think there's a missing time.
Oh, you asked me to read it.
No, it's not your fault, Dickson.
It's
I'm
the
just-
way it's written, but I
After
think...
taking time.
Fine, I'll start again. Zachary writes, after taking time to watch your iTunes U lecture, reading your textbook for class,
After watching.
and more recently, listening to your podcast on local meteorology and virology, I'm not going to get this job, folks,
You know,
I can tell
you're
you
not going to
now.
read any more letters.
I can't. It's the cold. It's interfering with my ability to read, I'm sure. I came across TWiV 190. I'm a little behind, in parens. In it, you put out the call for computer-ready volunteers. I'm a senior at Sarah Lawrence College, and I'm looking into starting a virology club on campus. If there's anything left that you can put a volunteer to, I'd be more than happy to volunteer in any way I'd be useful. Come September, I'll also hopefully have the membership of a small virology club to offer as well. Thanks for the education, Zachary. Perhaps they can transcribe the other missing TWiV episodes.
I might have some other jobs for them. Thanks,
Ah.
Zachary. And,
That's very
I'll be in
nice.
touch.
Very nice.
Yeah, you want to lie on the couch, Dickson?
Why? Are you going to analyze me?
No, not at all. That would be just a
No, that-
worthless episode.
No, it was a joke because you're a virologist. You can analyze what cold virus I've got.
Nah.
All right.
The next one's from Adam, who writes, "Ask and ye shall receive, or in this case, subscribe to citation alerts, and you shall be found, and you shall be found by cool papers. On a recent episode, you resolved to discuss more bug papers. Not long afterwards, this paper landed in my inbox, a drama seemingly perfect for TWiV. Among the cast are the industrious honeybee Apis mellifera, whose colonies are beset by an insidious parasite, Varroa destructor, carrying a devastating curse on its lips, or mandibles, I suppose. The mites unwittingly transmit deformed wing virus, DWV, to the developing larvae, leaving them crippled as adults. Enter Doctors Desai Yu, Wyhard, and Curry from the University of Manitoba, Canada. In this study, they test the ability of a DWV dsRNA construct delivered orally to protect bees from the effects of DWV.
I would think DWV would be driving while viral.
Yeah, that's a great name for an episode.
Okay.
We have to keep it in mind for a future one. Dixon's going to cough, right? You don't want to breathe into the mic? Thank you, Dickson. Wait, what's the driving while... What is the...
DWV.
DWI, right? Intoxicated.
DWI is intoxicated,
Okay.
yes.
At risk of spoiling the ending, it appears to work. The vaccinated bees form healthy wings, but the paper left me with unanswered questions, which I addressed to the Racaniello media empire. Very nicely written, though. Maybe you should go into writing, Adam. First, does this study actually demonstrate the effectiveness of dsRNA as a vaccine against DWV? The authors posit that an RNA interference response is occurring to limit viral replication upon later infection. I imagine that the RNAi response occurs in the first cells encountered by double-stranded RNA, the cells of the gut, rendering them non-permissive to DMV, effectively quarantining the gut. Assuming that DMV infection is naturally transmitted by the bite of the Varroa mite, local immunity in the GI tract wouldn't protect bees bitten later in life from infection unless siRNAs had been distributed systemically. The authors conclude from the lowered viral load and absence of wing deformity in double-stranded RNA-fed bees that siRNAs are being actively distributed. So this is a very interesting question that Raul Andino actually has worked on. And he has good evidence that indeed, if you say feed the bees or an insect double-stranded RNA, it actually the SI response becomes systemic. So,
And is that a specific response, or is it a generic antiviral?
I think it's
response?
specific. I think these are specific siRNAs that move systemically in the
Okay,
in the
because
insect.
I know a lot of there's been some controversy in the RNAi field medically that people are trying to use
Yeah,
this for
yeah.
new vaccine formulations, and there's some concern that there may be a non-specific component to the response that might not make it all it's cracked up to be.
Yeah, I mean, I could be misspeaking, but I'm hoping that Raul is listening in his car or while
Yes.
he's hiking, and
Vince,
he will.
do
weigh in.
insects have toll-like receptors?
Yeah, they do, Dickson.
So it's possible, just
they were
possible, then the double-stranded RNA treatment might be stimulating some innate immune response.
Yeah.
Right.
The real question here is whether it's a vaccine, whether it would protect them in the future. But I don't think insects live that long, right?
Bees don't live long at all. They don't.
So-
the queen does, but the workers don't.
Yeah, and I guess from a commercial standpoint, if you're talking about protecting commercial hives, which seem to be affected by this,
Yeah.
you'd be regularly feeding them stuff anyway.
That would be the idea, I think. So
Yeah, you
it's-
could just leave a little saucer of this stuff sitting outside the hive all the time.
Yeah, so I don't think it's a vaccine as much as an antiviral, really, right?
Yeah.
Right.
Because I don't think there's any memory in the innate system,
Ah.
as far as I know. Next, could the lowered viral load and preserved wings instead be the result of gut-localized RNAi stifling viral infection long enough for the wings to complete their development?
Great question.
Yeah. For example, having only 1% of gut cells remaining permissive after treatment would be equivalent to reducing the inoculation dose by 10 to the minus 1. I mean, that probably is partially the mechanism, right?
Exactly.
Yeah.
Perfect. You don't have to give it all the time. You have to give it just until the wings are developed,
Mm-hmm.
and then after that, you're set.
And finally, RNA isn't a super stable molecule. How would you expect to deliver double-stranded RNA-laced sugar solution to colonies of bees in the field? Are the chaperone proteins that stabilize RNA at environmental temperatures are there? Further, if the larvae are the vulnerable stage, double-stranded RNA would need to be consumed by workers, regurgitated, possibly stored as honey, and then consumed by larvae, all without being digested or degraded. Is that right, Dickson?
Well, I think so. I'm not a bee expert, but I think what he's saying has some validity.
Yeah.
The honeybees eat their own honey. That's their source of food.
Right.
So, if you can get it to them in these sugar-laced solutions outside, double-stranded RNAs are very difficult to degrade, you said in another episode.
Yeah, I think they're.
Yeah.
I mean, they're much more stable than single-stranded RNA, certainly.
So if
Yeah,
you
and
overload...
there's also some hocus pocus you can do to
Oh yeah,
RNAs
right.
to stabilize them more.
Right.
They also, in this experiment, fed the larvae with the double-stranded RNA. And so if the larvae will eat it that way, it doesn't seem like it has to be regurgitated.
Yeah.
I mean,
Right. That's right. It only has to do with their development. Do they know at what stage of development the crippled wings develop?
Whenever the wings develop, I would assume.
Well, I mean...
Which isn't much of an answer, is it?
No, not at all. As a matter of fact, I think they start as wing buds very early on during embryogenesis, but I'm not sure when this deformity would be absolutely interferable with, if you could stop it, like say at week 2 after development, then after that it doesn't matter, and that sort of thing.
Yeah, I mean, I assume because it's deformed wing virus and not absent wing virus, it's
Right.
probably post whatever the disc formation is that
Yeah,
does that.
that's right.
Looking at how they fed these bees here, how would you feed them? Where would you put the?
You'd put it-
This is done in the laboratory, I presume, right?
Yeah, but you'd put it in a little saucer with some sugar.
In the hive.
Or next to the hive.
Yeah, because aren't the workers feeding the larvae?
Yes.
Right.
Yeah.
So you need the workers to pick it up and take it in as food.
After 3 days, the freshly hatched larvae, less than 24 hours old, were grafted onto larval food in 24-well cell culture polystyrene plates.
Grafted onto it, yes.
Nice.
Yeah,
Sounds like a scene out of Alien.
right.
Okay, so they just put them in the food.
Yeah.
Right.
Just submerge them in food. That could be good, if it was chocolate or something.
Haven't we discussed a study where they did field feeding of
Sure.
insects with RNAi? I seem to remember one.
Yeah.
So I think that's been done, and the idea, you would put it out there and near the hives. Anyway, Raul, let us know what you think about this. The paper is Reduction in Deformed Wing Virus Infection in Larval and Adult Honeybees by Double-Stranded RNA Ingestion. It's published in Insect Molecular Biology.
Ooh.
I think I have to add that to my RSS feed because I don't really see anything in that journal typically, and this would have been a good one to pick up.
Yeah.
Right.
Adam, by the way, is in Melbourne, Australia, where it is currently 11 degrees Celsius and overcast.
Nice.
As of the time he was writing.
And who knows when that was.
That's right. That's right.
I should put a timestamp on these when they come in. I think it's just great that people tell us the weather, don't you?
Sure. Sure.
Alan, you are next.
Sure. Todd writes, I remember one TWiV that talked about how dry conditions cause mosquitoes to tend to bite humans more.
Right.
Are the current drought conditions in Texas maybe a factor here? And
Yes.
he links to a story on the BBC, Dallas in state of emergency after West Nile virus.
That's right.
And he adds, by the way, BBC has better coverage of America's news than the American news change. That's Shane, that sucks.
That's right. The answer is yes. I mean, this is a clear-cut case.
Yes, the BBC is clearly better.
And
sorry,
the U.S. news sucks, but so do mosquitoes in that case. There has been a huge drought in the midwestern portion of the United States now since last January. And if you look at the Drought Watch page from the USGS, you can get the archives for last year's weather, and it's color-coded, and the darker the brown color, the more the drought. And it just sort of hunkered down over Kansas, the panhandle of Oklahoma, and the northern part of Texas, just where the Dust Bowl originated during the 30s. And indeed, that's where the biggest outbreak of West Nile occurred this year, the epicenter being Dallas. And Dallas really got nailed. It had over 1,000 cases, and they had a lot of neurologic damaged cases as well, with some deaths. So this was this was the third worst year for West Nile virus outbreak in the United States, and it's all been drought-driven. So the mosquitoes involved in this are they survive very nicely during those periods because they breed in water that we create by our sloppy behavior. So we create these pools of polluted water, and then they come in and take advantage of it.
Right, and even in a drought.
Sounds like a self-fulfilling prophecy to me.
Right,
I mean, it really
even
does.
in a drought, we're creating these little pools and
Of course.
and such.
Well, I mean, every time you go out to the cemetery, for instance, I mentioned this on the West Nile story part, the cemetery has these little pots that you put flowers in to commemorate your loved ones, and they fill up with water during periods of rain. And then as the water starts to evaporate and the nutrient level goes up, there are odors that are given off that mosquitoes can pick up, and particularly the culicine and particularly the culex, and they go right for these little pots. And can you imagine going to the gravesite of your loved one that died of West Nile virus, let's say last year, only to be bitten by mosquitoes that were carrying the virus this year? I mean, there's a certain irony in all this. And so they identified all these places like sewage treatment plants. It's huge, huge breeding sites for mosquitoes, of this particular mosquito. So the answer is, even though there are droughts, you can find places of polluted water wherever you are. And that's how this virus infection makes its living, by taking advantage of that and the mosquitoes that carry it.
We need some antiviral schmutzdecke.
There you go. It's probably right.
Dickson, you need to enunciate.
I'm not enunciating.
Remember our listener on TWiP, he said we don't enunciate enough.
Really?
Yeah, you're kind of slurring there.
Am I really? You want me to repeat all of that?
No, I don't.
I don't either.
No, not at all.
No,
But thank you
thank
for
you.
your erudite analysis.
I was trying.
I thought you would like that question.
I love that question. Sure.
Kathy is next.
Robert writes, Doctors et al. A.
I think I was on a paper with him.
Really? That was Dr. Adol.
A. Love M, P, and V.
Nice.
By that, he means TWIM, TWIP, and TWiV.
There you go.
Each teaches and entertains. B. Could an episode of each show be a dictionary of terms to explain the science a little better for those of us who aren't in the field? I, who am a horticulturist, understand parasitism better than microbes or viruses or phage.
Cool.
C. Please relate more of those historic stories which give more life to the discovery of trich, giardia, or bubonic plague. Forever indebted to all hosts and hostesses on each show. Thank you.
Oh, that's great.
Dickson,
That's a marvelous idea.
what did the guy call you? The
Please.
a world class raconteur.
Yes.
We had a letter on TWiV the other
I thought it was
day.
a world-class train wreck. That's what I thought he said. But
A world-class parasitologist and a raconteur without peer.
I am flattered.
Do you have any stories for today?
Not on this topic.
Okay. Do you know Dr. et al.?
I do very well. Very well. I was a large part of it all, as a matter of fact.
All right, Dickson, I know it's your turn, but I want to take this.
I think Dr. et al. and Professor Staff are.
Yes,
probably-
that's right. That's right. That's exactly right. No, you can take this one, please.
This
do.
is long, and I know you're breathing laboriously, so you can take the next one. Okay?
That'd be great.
This one is from Alice, who writes, FYI, from the NWRA Wildlife Rehabilitators newsletter.
Wow.
What's the NWRA? National Wildlife Rehabilitator Association, I bet.
That would be correct.
So
good.
these are for people who go out and take wild animals who need help, right?
Yes.
Cool. The first
Wild
one,
animals who need to be in a 12-step program.
first is the new human phlebivirus discovered in Missouri. And that, of course, are these two farmers who got ill after tick bites, and they turned out to have a novel phlebivirus. We talked about on TWiV 199.
Yep.
So that's a cool story. The second is record number of human West Nile cases,
Right.
which we discussed. So we'll leave that. And there are links that you can follow. And finally, the third one I had not seen. Birds to humans, Usutu virus in Germany. Originating from Africa in the summer of 2011, Usutu virus is the cause of mass die-offs in blackbirds in southwest Germany.
Gee.
And again, this summer, thousands of blackbirds have died. Was found in Culex pipiens mosquitoes in Germany, and the virus may be transmitted to humans through a mosquito bite. First appeared in Europe in Vienna 2001, included some deaths in great gray owls in addition to blackbirds. In 2009, 2 immunocompromised Italians became infected. First human case of Usutu virus infection in Germany has been confirmed.
Huh.
But the affected man has experienced no symptoms of illness.
What kind of a virus is Usutu?
It's a flavivirus.
Well, it would have to almost fit into that group, wouldn't it?
Why is that?
Well, because it's transmitted by Culex.
Yeah, a lot of them are, aren't they?
Arbo.
Yeah.
Yeah. I just like this phrase, immunocompromised Italians. For some reason, I like the ring of that.
It has a good rhythm to it.
So that could be interesting, Dickson, right?
Very. Very, because I mean, if you hadn't said it was Usutu virus, it almost looked like they were describing another outbreak of West Nile.
Yeah.
It actually, so there is a page on ProMed Mail talking about this outbreak.
So is Usutu a place?
I assume so.
And if so, where?
Africa.
I bet it's Africa.
I mean, where in Africa?
Identified,
South Africa.
Is it?
yes.
Okay. Because the West Nile virus is from Africa, too, of course.
Identified 1959.
In 1959. Interesting. Cool.
I was 6 years old.
I'll be darned.
You didn't need to know that, I know. So, Dickson,
Yes.
it's infecting blackbirds
Right?
and gray owls.
Right.
It's transmitted by mosquitoes. So
Yep.
what is the reservoir? The birds?
Well, no.
Maybe some other animal?
Well, the reservoir, it depends on who you are. How are you speaking? As a human or as a bird?
He's enunciating.
I'm speaking with a forked
A forked tongue, that's snakes. And I don't think snakes are reservoirs in this case.
He's speaking as an immunocompetent Italian.
I am.
That's right. That's right. I've just returned from Italy, and I am now an immunocompetent speaker of Italian.
You can say I don't know, Dickson.
I was going to say, I don't think it's discernible from this description.
Right? Is it the bird? Not necessarily, right?
No, that appears to be the definitive host.
What does that mean?
That's the one that catches the infection routinely.
I see.
And the animals die off. That would seem to not be the
Reservoir.
reservoir.
That's correct.
Yes, that's correct. So maybe it's some other wild animal that the mosquito is picking up the virus from, because they like to bite blackbirds, right?
Well, maybe other birds serve as reservoirs because they don't die.
Right.
Just like West Nile. In fact, West Nile,
Yeah.
in Africa, the birds don't die anymore.
So before West Nile, I didn't realize that mosquitoes bit birds.
Oh, my gosh, yes.
It's interesting.
That they wiped out 5 genera of birds on Hawaii.
Who the mosquitoes?
Yeah, the malaria that they brought in.
Oh. Wasn't malaria first described in birds?
It was.
Canaries?
It was by Sir Ronald Ross
Ronald Ross.
in India.
He was
correct.
He was a Brit, right?
He was a Brit. It still is.
He still is a Brit.
Well, you
He's
you're never
he
not
doesn't
a Brit.
exist anymore.
That's right.
Dickson, you can take the next one from Jim.
I will be happy to do so, and in doing that, I will try to pronunciate every single syllable. Vincent. Gee, that leaves the rest of us out. I'm going to stop right there.
Okay.
Vincent. I'm working through some British podcasts, 2 of which concern evidence-based medicine. It seems that most work in this area is done in Britain, Canada, and Australia. These are the podcasts. Evidence-Based Medicine, a 43-minute lecture, and Future of Evidence-Based Medicine, a 30-minute lecture, both by Professor Paul Glasziou. The Evidence-Based Medicine journal mentioned is I'm not doing very well here, am I? The Evidence-Based Medicine journal mentioned in one has a lot of RSS feeds that I like, and the Mendeleev service has a virology group. Should virology, microbiology, and parasitism be a part of this concept, or have a similar program? A good PowerPoint presentation notes, as does another of these podcasts, that over 30,000 clinical trials are started each year, which must affect every biomedical field. This is the first time I've seen how big the research field has become. Is this a major reason that grants are more difficult to obtain? Is the upward trend in numbers of trials likely to continue, or is the system beginning to break down or evolving into something else? TWiV 197 with Professor Marcus was terrific. Well done and just right on target. I hope you can capture any other senior researchers who have had seminal roles. Thank you. Regards, Jim Smithfield. No, regards. That's Jim Smithfield is the next letter, I guess.
I captured Dick Despommier. He's a seminal researcher.
Well, when he can read properly.
He's certainly senior.
Yes, he's having his senior moments right here.
Alan,
Yes.
you must know about evidence-based medicine, right?
Oh, yeah.
Tell us, give us the executive summary. What is
Right.
this?
It's a movement to get better scientific basis for more of medical treatment protocols. A lot of, science has always had this rigorous approach to you've got to prove something, and everybody's got to try and falsify it, and then it stands. Medicine kind of grew up in a more organic way, and in many branches of medicine, treatment is very empirical. It's just, well, we do it this way because we do it this way, because it worked in the past, and so we're going to do this. Infectious disease, not so much. There you have specific drugs to treat specific pathogens, and you even have mechanistic insight in many cases. But if you're talking about something like, cardiology or pain management or certainly psychiatry, there are many, many branches of medicine where it's very vague, and you look back at the evidence, and if you dig into the literature, you realize, well, this might just be folklore.
Right.
So evidence-based medicine is this idea that we need to go and test these things and up to modern standards. It's kind of bringing everything up to code in medicine.
Yeah, a good example is the former treatment for ulcers.
Yes.
Where they would classify you as a type B or a type A personality. And type A people were, of course, going to get ulcers until it discovered that it's caused by a bacteria. Oops.
Yes.
Yeah.
And that's the evidence that kicked out that stuff. Yeah, that's right.
He asks whether the growth of that field is one of the reasons why grants are more difficult to obtain, and I think it's much more complicated than that.
I do too.
Well, clinical trials are mostly that 30,000 clinical trials figure. A huge proportion of that, I don't know exactly, but a huge proportion has got to be industry-sponsored trials.
That's right.
Mm-hmm.
That's right.
So that's not the grant crisis that we've been talking about, and the structural problems that affect academic science. These clinical trials are being conducted in most cases at academic centers, but they're run by industry in support of a drug application in most cases, or a device application.
Yeah, the thing that's wrong there, and I know you know this, Alan, because you've talked about this many times, is that the evidence that gave rise to the fact that they want to test this drug to begin with came out of that industry. And so, and you look at the evidence that caused them to say, we have enough evidence now to want to do a clinical trial. And many times the evidence was rather weak.
Yes.
But of course, the drug company has lots of money, so a lot of medical schools get suckered into participating in these larger trials where the drug itself has not proven out to be very useful.
Yeah, and if we want new drugs, that's the system for doing it, unless we're ready to have the government take a bigger role in clinical trials,
That's
which
right.
which people have advocated, and I know the NIH has made kind of a preliminary move in that direction with this translational medicine initiative. But that gets into the whole issue of how much public money are we going to put into clinical trials?
Mm-hmm. Yeah, and Columbia University does a lot of them.
Of course, yeah. All the big academic medical centers do a lot of them.
Yeah, they make.
And it's not just a money issue. This is where new medicines come from.
Sure.
Mm-hmm. Yep. Okay. Alan, I believe
Okay.
you are next.
Sherry writes, "Hi, Vincent et al. I'm writing to ask if you would address the recombination potential for live attenuated influenza viruses in the flu mist vaccine. I enjoyed TWiV 191 when two rights make a wrong, but was left wondering why recombination of attenuated flu viruses wasn't considered to be a risk. Thanks in advance. All the best, Sherry. P.S. I'm a second-year PhD student in the University of Maine Oceanography program, conducting research on persistent virus infections in marine phytoplankton and virus-mediated gene and carbon flow among marine trophic levels. I'm working in Willie Wilson's marine virology lab at Bigelow Laboratory. Very cool.
Nice is right.
Yeah.
That's great.
Thanks for transforming my long commutes from a chore into a cherished part of the day. My time to listen to TWiV. Gosh, a long commute in Maine. That's got to be. And Vincent, you wrote to Peter Palese.
Mm-hmm.
Do you want me to read his
Yeah,
reply?
sure.
Okay. So Peter writes back. Peter's obviously a huge guy in flu. And he says, this issue has been discussed at length before FDA approval, but there were no concerns raised by the FDA during the hearing. Additional points. 1. The HA and NA on the vaccine strain are those of co-circulating human strains, or at least those from strains circulating a year or two earlier. Thus, nothing new would be introduced into the human population where were those HA-NAs to escape. 2. The non-HA-NA genes of flu mist, in essence, all contain attenuation markers, temperature-sensitive, cold-adapted, etc., and thus would make the circulating wild-type strains less fit when reassorted into them. And 3, the M gene of flu mist has an interfering activity, and thus reassortment is actually pretty difficult when a CA strain is involved. Julie Youngner from Pittsburgh has or had a patent on this therapeutic effect of flu mist. When flu mist is given to a patient, this patient appears to be protected against a super-infecting wild-type influenza virus for at least a week or two.
By CA, he means cold-adapted. So reassortment is pretty difficult when a cold-adapted strain is involved.
Yeah, so that's what the flu mist strains are. They're cold adapted, among other things. So I had sent the question, do the flu mist strains, is it a risk to reassort with circulating strains? Because reassorting among themselves wouldn't give you anything.
Right.
You couldn't get any complementation by that among the cold adapted strains. So apparently there's no risk. So there you go from the boss.
Huh.
Okay, Kathy, you are next.
Frank writes, "Dear wise men, in chorus with your many
The end.
listeners..." Yeah, it's all right. I don't take offense. "Thank you, and know that we depend on your continued TWXXing." So that's Twin, TWIP, and TWiV again. "I was lucky to have stumbled upon TWiV in the single-digit era and have not missed my weekly TWiV inoculation since. After 40 years of building technical materials companies in many markets, I am trying to turn my energy towards societal goals, but you guys make it very hard to stay out of the commercial implications of the weekly microbiome revelations. Oh, to be 30 years old again. I've just finished Vincent's pick from number 192, Microbes and Evolution, and a paper referenced in the Rauer chapter, number 9, on phage, which describes the broad presence in phage of exotoxin genes common in pathogenic bacteria. I can just imagine bacteria paraphrasing Flip Wilson, who said, 'The devil virus made me do it.' And then he gives a link to that paper. I would like to suggest that you consider an episode dedicated to a paper such as this and the topic of virus as the ultimate and continuing force behind evolution, sustainability, and life. Thanks again to all of you for giving your time to share your knowledge in such an accessible style and vehicle. Best regards, Frank. New York, New York.
So we had actually picked Forrest Rower's book on TWiV 87. It's called Coral Reefs in the Microbial Seas. It's really good. You might enjoy that, Frank.
Yeah.
It's all about... So Forrest goes out on boat trips and samples. He's interested in what's making coral reefs decline. He has some very interesting... Actually, we should cover one of those stories. You know, there are herpes viruses of coral reefs. And
Yep.
It's very... You know all about this. When you get runoffs, right, the coral reefs suffer and all of this.
Sure, and the sea levels are rising, and the temperature at
Do you
which...
really believe that?
After Sandy hit, yes, I certainly do. But there are other non-microbial effects on coral reefs that we should pay attention to.
Kathy, I think this came in before you joined us, actually. So I'm sure
Okay.
Frank would have said, "Dear wise men and women."
I'm sure. Yeah.
Sure.
Not a
And
problem.
being a New Yorker, Frank, you know, he's savvy.
Yeah.
I will take the next one from Len. I would like to hear you discuss this on TWiV. Flu shot linked to higher incidence of flu in pandemic year as a CBC story. It's about work presented at an ICAC poster session by Dr. Danuta Skronowski et al. They say it might be related to the dengue fever phenomenon that you've discussed before. Antibodies to one serotype increase the risk of serious disease from a different serotype. In their experiment, they gave influenza to ferrets, but don't worry, it wasn't H5N1. It was only the 29 H1N1 pandemic strain. So there is a video of this presentation at ICAC on Microbe World, and I'll put a link to that in the show notes. We did discuss, so the observation here was that in Canada, and Alan, you probably remember this,
Yep.
in 2009, people who got in the previous year who got a seasonal vaccine, some of them fared worse in 2009. And it was just in Canada, you know, it was labeled a Canadian thing. Weird, no one could explain it. And then now what Skronowski has done is to do an experiment in ferrets where they immunize with the 2008 seasonal vaccine and then challenge with the 2009 pandemic strain. And indeed, some of the ferrets get more serious disease. And so there's no explanation except that maybe you get some cross-reactive but low-affinity antibodies from the immunization. And then when they interact with the pandemic strain, they make disease worse, sort of like dengue. Second and third antibodies make that infection worse as well. So it seems to be a real phenomenon, but they don't know why.
Well, it may be a real phenomenon. It's not clear why nobody was able to find this signal in any of the other flu monitoring from other countries.
Really, eh?
So that's why it was referred to as the Canadian thing, because it doesn't show up, as far as I understand, it doesn't show up in the CDC data. It doesn't show up in anybody else's data. So it was kind of a fluke thing. And then this ferret experiment, I mean, it's suggestive, but it doesn't really get you back to the issue of does this really happen in people, and did it really happen in people? Yeah. But it's peculiar.
I would not put a lot of faith in the ferret experiments.
You mean ferrets are not people?
No, they're not. I mean, to explain something that you observe, as Alan says, in one population by a ferret experiment, I think is very difficult. She says
it bears looking at. It would be very interesting, since this hadn't been observed with any other vaccine in the past. You know, maybe this is something that's unique to pandemic years. Maybe it's unique to that particular vaccine. There could be some interesting immunological insight in there.
She makes some pretty strong points about that in the video, and also points out that the kind of experiments you need to do are challenge experiments, and you can't ethically do those in humans.
Right.
So ferrets is the next best thing.
Of course, yeah.
Yeah, she's very good about explaining that, and also saying this is an interesting observation. We have to follow it up.
Yeah.
But why it would be only in Canada is very puzzling, right?
Yeah.
I don't know.
Or it may just be that the Canadians kept better data. I don't know. But I find that a little hard to believe.
Yeah. Dickson, the next one is from Shinai.
Yes. Hi guys of the TWiV. My name is Shinai. I'm biochemical virologist and illustrator in Brazil. I like a lot of your work. I can say that every week I've always watched the news from you. I love science, virology I'm passionate about, and I am one of those who believes that viruses were the first living and viral symbiosis in humans, which today is already a fact, but some years ago I was called crazy because of it. I'm reading this verbatim here.
Well, it's fine, Dickson. I'm just laughing, not at you.
No, I understand. Look, I try Portuguese. That wouldn't work. Well, I'm creating paper models of papercraft for science and did a bacteriophage, like origami, and an HSV1 that serves to decorate or to help and educate. I wondered if you'd like me to put my designs on TWiV page. It would be an honor for me to have my paper crafts and graphics on the page of yours, and still allow each virologist might have a virus on your preferred model home. Contributing, we could reword this. Contributing to the advancement of science. I know what she's saying, though, unless that's not a she. So this person has taken up the job of representing, I guess, viral shapes using origami, like paper models. Yeah. Does this person submit any so we can look at them?
Yeah, there's a Facebook page. Yeah, they're kind of nice.
Right, and how are they?
They're nice. There's a page.
So let's put them up.
Yeah.
Let's do this.
Yeah,
and for those of you who aren't on Facebook, you don't have to be on Facebook because I'm not either. You can just go to the page and scroll down until you see things that look like viruses.
Oh, wow. Look at that.
Yeah, and there are a lot of, it's not just viruses. There are a whole bunch of science papercraft things here.
Stomach, kidneys.
Yeah.
Nice.
Liver, eyeball. It's cool.
Terrific.
Right,
yeah, sure, absolutely.
We'll use your illustration for the episode art. How's that? I forgot your name here. How's that, Shinai?
Shinai.
Yeah.
Interesting. Very nice.
I vote for the phage.
Aha.
Okay, we'll do the phage. I liked that page a while ago.
Mm-hmm.
So let me get this straight. From a page, she made a phage.
Ooh, Dickson. What a great one.
Nice.
That could be a title, too.
Could.
Yes.
Could.
From a page, she made a
phage.
How about from a page, a phage?
Right.
Yeah.
It's all the rage.
Then it's New York Times style.
Hey, guys, I noticed that Rich Condit has joined
Really?
the document.
He's joined the document. You think he's going to dial in soon?
I don't know. Just
maybe
be prepared.
Maybe it's his plumber. It'd be nice to have a plumber on TWiV, don't you think?
I do think so, absolutely.
No, they'd be coming out with a lot of cracks.
See, we set you up, don't we? Dickson did that last one.
I did.
I think it's back to Alan.
Okay. See, Jim writes. Oh, we're back to Jim.
Yeah.
Jim writes a lot.
He does.
No, it's great.
Jim is cool. He listens to a whole bunch of podcasts and draws our attention to a bunch of stuff that we probably would have missed otherwise.
There you go.
So he says, "Hi, Twivers. Thanks for pointing out the use of paper flicks in session 198. New concept for me. I see how the accordion concept was conceived. Paper flicks will allow multiple poster presentations at conferences by a researcher involved with several topics. It looks like the start of another mini industry. Your mention of Khan Academy reminded me to send this link for an interesting Bozeman site that has some 70 video segments dealing with biology. I don't know how one teacher could put so much together, but it's the best collection I've seen. Hey, the TWiV page has over 200 presentations. All right. Here's the 10-minute, 30-second video on viral replication so you can see how well it's done. For some reason, it is one that shows a distorted picture, though the timeline mini shots are readily seen, and a strange ad at the beginning was clear. So there apparently a few little production glitches, but some.
But just looking at the page, the flash page of all these videos is amazing. This
guy got the Montana Teacher of the Year in 2011.
Goodness.
And I'm not surprised. I mean, it's everything from data analysis to viral replication and ecological succession and huge range of topics.
Apparently, this guy did a TEDx talk, Dickson.
Really?
Yeah.
Nice.
Good.
It's cool. I like that very much.
Yes.
Rich put one down here that he likes. Yeah, this is a very cool one. It's called And since Rich isn't here, it is called Reflections on Digital Aristotle. It's very cool.
Yeah.
Check that out. That's good. Thank you, Jim. I would never have found that. Kathy.
Seamus writes, "Hello, Dr. Racaniello et al." There's et al. again.
Yeah.
Tell you.
I saw this paper and thought I would pass it along. The authors present data for the inclusion of viruses in the tree of life. Since the question of whether viruses are living or not is a common topic of discussion on TWiV, I thought it was worth sharing.
Right.
I really enjoy all the TWiV podcasts and appreciate all your efforts. And so he sent this paper, and then we have a couple of related follow-ups. But I loved it because I went to the paper because I thought maybe we're finally going to see a tree and we're going to see viruses on it. And we do. So it was quite exciting. So he's got the Biomed Central link there for the original article. So it's open access. People can check it out.
This is a cool story where they basically make phylogenies of proteins from these big viruses like mimes and et cetera. And they find that they can make a separate tree, archaea, bacteria, eukarya, and these viruses. And they make the suggestion that these viruses arose by gene reduction, basically, because they can see the loss of certain viruses in these big DNA viruses, and they suggest that they're involved in also mediating gene transfer among cells. So yeah, you could put. I mean, these are all pretty big viruses, and they're all DNA-containing viruses. What about the smaller guys? How do they fit in? I mean, these big ones are very interesting because they have genes that nobody else has, And so where did they come from? Right. You don't find them in cells, so where did they come from? I don't know. It's really interesting, though, so it's a good read.
Comets.
Were you at the University of Georgia, Kathy, or were you at Georgia State? You were.
No, I was at UGA. Mm-hmm.
So they have that's the veterinary medicine. Okay.
The vet school.
Okay.
Cool. Well, so that settles it. Viruses are alive.
Well,
let's read the next email.
yes.
Moving on.
It's related.
Yeah, it's from Magda. Hello, TWiV team. I'm a student from Poland. This year I'm beginning my master's in biotechnology. First of all, I would like to thank you for this podcast. I haven't been attending to any virology course yet, so most of my knowledge about viruses, since now, is from TWiV and my own researches. When I started listening to TWiV, I had to check many things which I didn't understand, but with every next episode it was easier, and now I can say that I learned a lot and found viruses really fascinating. So she then gives us this link to the same article, that, or actually a Science Daily summary of it, about the same article. And she gives some quotes from it. I would like to know your opinion on this subject. I have already heard in one episode that you believe that viruses are not living organisms, but just chemical molecules. I've been thinking about this for a long time, and I do not understand, why do we have to call something alive or inanimate? Isn't there something in between? Why do we have to describe something as black or white? What about gray? And why isn't there such a problem with parasites, which also cannot live on their own but need another living organism? I know you're getting lots of emails, but I hope to hear from you soon. Waiting for the next episode, Magda. P.S. I'm one of those who listens to TWiV till the end. This is an old email, actually. Well, isn't there like living and dead and nothing in between, Dickson?
Well, wait a minute. I'll ask the guy sitting behind me.
There's nobody behind you.
It's not fair to raise this question unless you place it in context with other biological functions. Replication is just one of many, as we discussed before. Viruses have no innate metabolism of their own, whereas most other organisms do. So I think that's the break point. The break point is there a metabolic pathway associated with a virus? And the answer is no.
So you think viruses are not living?
Do you know of any?
Well, not in the particle, of course.
Not in their genome. There's no
genome is just DNA.
There's no energetics in their genome. None. Zero.
There's no metabolism in the particle.
So therefore,
as far as we know.
How could they possibly, if we gave them everything they all needed, how could they possibly make it on their own? And the answer is they can't.
No,
Well, they can't make it on their own, but it depends on how you approach the question of whether something's alive.
right.
And there's sort of a trend in ecology to start looking at organisms not as this species is species X and this is species Y, but to look at them as continuous functions. So in other words, defining an organism as what it does. This is the organism that fills this particular niche and has these functions in the ecosystem. And viruses certainly fill particular niches and have functions in the ecosystem.
Well, so do transposons, but you wouldn't call those viruses. They used to be, maybe.
Maybe.
It's a piece of DNA.
It's a piece of DNA, that's right.
It's not is DNA living, Alan?
It's certainly part of life.
You dodged the question.
That's right. You'd make a good politician. No, really, I think metabolism plays a big role here. And I think if you don't define life as metabolic, then you're stuck on these issues of whether it's alive or not. So my definition of being alive means that it has to show some sign of life. And the sign of life I use is a product of metabolism, either CO2 evolution or oxygen evolution or sulfate, metabolism or something.
But viruses have all of those things when they're carrying out their normal living functions.
In the infected cell, you mean?
Yes. The infected cell.
An infected cell is living. I would not debate that at all.
Nobody does.
But the particle itself is
the
not
virus
living.
is living within the infected cell.
It's taken over the whole cell so that it's now a virus-infected cell, right? But the
So
particles that are in it aren't living. The virions.
You know what it really did? It assumed some portion of the host's genomic function.
Yeah,
and subverted
none of the individual components of the cell are living either.
And subverted it. Oh, that's not true, Alan. Come on, you're wrong on that.
The cell's DNA.
No. I'm thinking about mitochondria and chloroplasts. They probably were derived from other life forms before they became symbionts.
So where they came from determines whether they're living?
No, but they have a genome and they have metabolic functions and they produce things.
Yeah, and I think viruses have genomes and metabolic functions too.
I don't think they have metabolic functions. They take advantage of metabolic functions. That's what they do. So they're subverted expressions of hosts' genomes. That's what I think they are.
So, Alan, you believe viruses are living, right?
I lean toward living, yeah.
You do.
I mean, the real thing is here, Magda, I think the whole point is the discussion of whether something is alive or not. You just get places when you discuss it.
That's true.
It's just labels that we put on things, and it doesn't really matter.
Yeah.
But it's for learning purposes.
But if you defined life in a certain way, then you can exclude it. But if you define it in another way, then you can include it. So if you say that parasites can't survive without their hosts, I would agree. But I don't know of any parasites that don't have metabolism.
And the discussion highlights areas. Some important issues in biology. We put these categories on life, and life didn't get the memo.
Well, I hate to say this, but every time we shoot a rocket off to Mars, we're looking for life as we know it. We're not looking for life as it could be. We're looking for life as we know it, and that's billions of dollars set up on the premise that life has metabolism associated with it. That's what we do. So
don't you wonder what I think?
Yeah, I'm waiting for these guys to stop. I was going to ask you.
This was addressed to gentlemen.
No,
I was just joking. Just joking.
I don't think we've ever had Kathy's position.
Kathy, that's right. How do you?
So, I think having this discussion is really useful because it makes everybody think about what is life and what is your definition of life. And I think that when the virus is in the infected cell, I think it's living. I also agree with Magda that why do we have to have black or white? Why can't we just have gray? And most of the time while you guys were just going on now, I'll listen again when I listen to the episode. I was just thinking about the physicists and is light a particle or is it a wave?
And the answer is yes.
Yeah. So that's kind of where I am on it.
Really?
Dickson, or Kathy, I hope to enter the gray area and not the black area. Okay?
You think this is a quantum-like question?
You know, one of the most popular pages on my blog is one called "Are Viruses Living?"
Huh.
It gets the most hits of anything.
Yeah.
People are really interested in this, and I think it makes for good discussion.
Yeah.
But I don't think you should say you're wrong or you're wrong. Just this is how I feel, and this is how.
Right.
Exactly right.
There's no answer, right?
There is. It depends on how you start your definition. If you use metabolism as a definition of life, then as NASA does, and they do, that's their definition of life. It has a metabolism.
Yeah.
So therefore, they
if
go out
there
and
were
look
a
for
clear
it.
answer, this would have been a much shorter discussion.
That's right.
By the way, the fact that you can put these big DNA viruses on their own tree, right?
Yeah.
And the reason you can do that is because they share some genes with cellular organisms, right? And that doesn't, of course, prove that they're living or not. It just shows that in evolutionary terms they can be there. There's one more on this paper from Hans. Dickson, can you read this?
I will try. That was a good question, though. Ken, not May. So Hans writes, Dear TWiVers, please see attached an interesting publication regarding giant viruses and their consideration as a distinct and ancient form of life. I personally disagree with the authors to consider viruses as a distinct form of life. But according to the author's conclusion, giant viruses have played an important part as messengers for gene transfer and contributed to biodiversity. This is a very interesting aspect and keeps the brain thinking. The manuscript he refers to is...
You don't have to read
the
that.
one we were just talking about.
Yeah, it's the same one we were talking about.
Is the same one we were just talking about. You let me finish.
Oh, sorry, Dickson.
And then he says, and there's more. Please.
I know, I'm difficult. I know.
Who has the spotlight right now? Come on. The guy with the cold. I really enjoy your podcast. Really great work. And I do recommend it to my students to listen to it. I am based in Melbourne, Australia, and teach and research at the Department of Microbiology, Monash University. Melbourne has actually 4 seasons, and now we are heading towards summertime. Let me know if one of you goes to Down Under sometime. Would be nice to catch up. We have definitely nice viruses here, such as Hendra. Cheers, and all the best, Hans.
His name is familiar.
You should let the Australian Tourist Board know about that.
I could have done that in an Australian accent. Darn it, I could have done it as the gecko.
The
Geico gecko.
Can you do an Australian accent, Dickson?
Well, I can without my cold, but
Hans Netter is a familiar name. Did anybody recognize that? I've seen it before.
Yes.
Yeah.
He has 4 seasons. What does that mean?
Winter,
spring, summer, fall, and winter.
Yeah, well, doesn't everybody have 4 seasons? No?
Well, just pointing out that they're heading into summer.
Okay.
Yes.
Bye, Dickson. Dickson's coughing.
Well, a lot of tropical countries have 2 seasons.
Yeah, I guess, yeah.
So.
I'm spoiled up here, right?
And it's just a different way for him to talk about his weather. That's all.
Yes.
Yes, of course.
And
Alan,
that was a good way to talk about the weather in an email, by the way, because whatever weather you say in your email will not be current months later.
Once we read it, that's right. We have 4 seasons. How you doing, Dickson?
A little bit better.
Right. I'm glad you came in just for TWiV.
Well, I'm glad I'm a living example of the way viruses affect people's behavior.
Alan?
Yes. Sarah writes, Hi, TWiV team. A terrific blog I follow had this post today about avoiding what they called predatory open access journals. I thought you all in the TWiV audience might find this heads up useful. It's a disappointing development for the field, but I suppose from an economic perspective, it shows that predatory entrepreneurial creativity is alive, well, and continuing to expand its range. Best wishes, Sarah. So this is it's a blog about hospital infection prevention. And this post is if you're involved in academia or if you do any kind of science writing, I know this for a fact. You get emails on a regular basis inviting you to be part of the editorial board or to submit your paper to our new journal, which is the Journal of Wada Wada Wada.
That's right.
And a lot of these, in general, that tactic is from these organizations that just pop up like mushrooms, and they are vanity presses essentially. If that, in most cases, they're just doing this for the page fees. So you would send your paper to them. They would say, "Oh, thank you very much. Your paper's been been approved by our editors when in fact they didn't even read it.
Right.
And we're going to publish it. Just send us your check for the page fees.
Exactly.
And
Nice.
that's their business model. And these, some of the larger ones, actually, I'm not going to name names, but you can find these very, very easily if you're not already getting spammed by them. Some of the larger ones actually do have websites where they publish this dross that they take in, and and in a lot of cases they list people on their editorial boards who have nothing to do with the journal. So it's a huge scam that's riding on the back of the open access movement.
The original article is in the Chronicle of Higher Education.
Yes.
It's a Q&A with Jeffrey Beall on predatory publishers. It's very good. And he talks about what, he thinks this is and what publishing should be. And this is very good. I'll read just this one bit. I support what I call platinum open access. This is open access without author fees and with the publication costs supported by volunteer work and benevolent funders. There are a few publishers that now use this model, but it's not sustainable and it's not scalable to all of scholarly communication. The only truly successful model that I have seen is the traditional publishing model. And we have talked about that quite a bit here on TWiV.
Yeah, so these operations are the gold open access model.
Okay.
It is also done by a legit journal. So PLOS One is a
Sure.
gold type open access business model. They use page charges.
Right. Kathy, we have reached you.
Okay. Let's see.
Renato,
yes. Hi, Vincent et al.
Here we
Again,
go.
thank you for your wonderful podcasts. I wrote you a little while back suggesting that you could hire someone to transcribe the shows and put the text online so that Google can index it. It occurred to me that some listeners might want to do that, so you could crowdsource the effort. So we talked about that at the beginning, and hopefully we'll get some more takers on that. I want to try to explain to you a few concepts in computer science because I wonder if there are natural processes that work in analogous fashion. I am not an expert in any of this, but I do study it. Let's pretend a computer program has two instructions. Go to the folder X in the hard drive and erase everything at the current folder. Notice that this is a legitimate program. That is, if I want to delete everything at folder X, I run this program. The actual program that is run by the computer consists of a series of numbers, which codify the steps to be actually performed. Let's say that this particular program is codified the numbers 01, 02. That is, 01 means go to folder X, and 02 means delete everything in the current folder. If I wanted to do some harm, I could devise a way to start the execution of this program at step 02 instead of at its start point. This would make the computer execute the instruction, delete everything in the current folder, wherever it currently is, which may or may not be the folder X. There is, however, an even more ingenious way to use the program list. To understand it, please notice that computers don't need spaces between numbers, so the listing 01 space 02 is stored in the computer as the list 0102. Then I could force the computer to start execution at the second digit. That is, I could make the computer execute the program 102 instead of 0102. Now, what does this 102 listing codify? It could be that the number 10 is an instruction understood by the computer. For instance, it could mean flash the screen once. Also, what happens when the computer reaches the number 2 instead of 02? Does it automatically insert the leading 0? Does it stay put, waiting for the next digit? I ask these questions to you because I imagine some transcription processes might face analogous situations. What happens if, because of some ribosomal error, a gene starts to be transcribed not on its first codon, but from some middle one? What if the mRNA loses a base somewhere? How frequent is such stuff? We can measure DNA replication errors because we can observe mutation, but can we observe transcription malfunction? Can we make it happen more frequently? Also, are there viruses that hijack cell functions not in a per gene basis, but in a dirtier manner by starting transcription of already encoded stuff in our genes that is shifted from their correct positions? For instance, say that the sequence AGA, AGA, AGA encodes a protein needed for the cell membrane. Say that the sequence GAA, GAA, which is encoded by the same sequence as before but shifted one letter, encodes a protein which is part of a virus capsid. Wouldn't it make sense for the virus to use our genome as a library, but reading the books using a different index than our own? I'm sorry for the long email and grateful for your attention. from a very hot and dry Sao Paulo, 34 Celsius, 61 days without rain. Renato.
Wow.
I think he means translation, right?
Yeah.
Yes.
Okay.
Well, the first time he mentions transcription, yeah, he does mean translation there. But you could think about some transcription errors, or maybe more appropriately, some splicing errors that could give you.
But what
frames.
if the mRNA loses a base? Then
Right.
that would screw
Right.
up the reading frame.
Right.
You want to weigh in on that, Kathy?
Well, I thought the computer analogy was quite interesting, but I'm trying to think offhand of cases where viruses might force ribosomal frame shifting.
I don't know.
Particularly of cellular genes. I can't think of it. I can think of retroviruses where to get the reverse transcriptase, at least one of them does it by a ribosomal frame shifting mechanism.
Yeah. So it happens in viral genes, yeah.
Yeah.
But I don't know of any induction of it in cellular genes. Where you have a hidden capsid sequence in a cellular gene and it gets out. I don't know of any example. I mean, in theory, you could do that, but I don't know of any example of it.
It's really creative thinking. I think it's a really interesting concept.
Yeah, I think there'd be enormous selective pressure for the cell to fix that.
Yeah.
Obviously.
Yeah, right.
Yeah.
But it reminds me of an old saying in computer science that there are 10 types of people in the world, those who understand binary and those who don't.
That's right. Right.
Yeah, I gave my brother that T-shirt, in fact.
Okay.
So the idea of the errors in messages that lead to translational errors, like starting translation in the middle of a gene, that's controlled for. That's why we have ways to make sure that translation begins at the right position. But I'm sure that sometimes it happens, but the proteins are defective, and we have very dedicated systems for getting rid of misfolded proteins, small proteins that aren't the right size, and that sort of thing. So, it happens, and people have quantified the translation rate. It actually is amazingly high, but there are correction systems that throw out the crummy proteins, and so you don't have to worry about them.
But yes, this does happen.
It does happen, absolutely.
And it is a phenomenon that viruses exploit in their own genomes, if not in the cellular genome.
If you can imagine it, it's real.
I don't have the number. I'll try and dig up a link to some numbers about translational malfunction.
Does RNA editing play any role in any of this?
Yeah, so some RNA viruses undergo RNA editing, so they'll put in an extra base or two and change the frame and make a different protein. Sure. I know if parasites do that, right?
Yes, quite frequently.
Mm-hmm.
Okay, I am waiting for Rich to join us. I know we are now at an hour and a half, and we will go a few more minutes because we have, we'll never get through all these emails. But Dickson,
yes.
The next one is from Etienne.
Etienne writes, "Hello, professors. I've been a long-time listener of your great podcasts. Since you traded a few of your favorite quotes about science at the end of TWiV 199, I decided to share one of my favorite. It's from Isaac Asimov and goes as follows. If knowledge can create problems, it is not through ignorance that we can solve them. I think it's a valuable quote to keep in mind when science and politics comes at a crossroad." That's a good quote. I like that quote.
This
Yes.
one. Yeah.
Yep.
Yeah, we're going to put that one over my door.
You have a door?
I have a door. It leads into your office.
The next one's from Ibrahim. Dear TWiV team, I have a problem that I want to investigate in TWiVo.
Good.
Yeah.
Very good.
It's like in vivo, right?
Indeed.
In TWiVo.
It's another title. I'm a graduate student that just finished my master's degree program in infectious diseases from Heidelberg University, Germany, basically working on viruses. I did my thesis on oncolytic viruses and also have experience in virus-host interactions. I became stuck to virology and willing to proceed further, doing also my PhD in virology. Now I feel myself lost in space, going through announcements for scholarships and open positions here and there, sometimes with hope of getting a position and others with desperation When it comes to applying for a scholarship, I think I face a problem with writing. I don't know how to write a good research proposal. I faced the problem earlier when I was writing my thesis that I am too slow and don't know how to start and when to stop writing. My question is, are there standard ways for writing in science that I shall follow as well as everyone, or is it just like art and related to personal attitudes and styles? I would appreciate any help in this regard, and keep up the good work.
Wow.
Wow. It's
So-
definitely not, just give me 1 sec, Kathy. It's definitely not like art, right?
No,
Right.
no, it's anti, as a matter
Right.
of fact.
Go ahead, Kathy.
So two things are going on here. We just got a picture from Rich of
Yes.
the plumbing stuff going on in his bathroom. So I was slightly
Oh, yes.
distracted by that.
You're not multitasking, huh?
No. And then, but the first thing that came to mind is that there are some classic books about writing papers, and the one that is now in its 7th edition is called How to Write and Publish a Scientific Paper by Robert Day and now Barbara Gastel. There are books about how to write grant proposals. The one that I'm most familiar with is not on Amazon, but directly on the website called grantcentral.com. So those are some things that sort of start you out in a formulaic way to start with an outline, start with a few, just write this sentence, now string these sentences together. That definitely happens in the grants workbook. And then, as Alan has just added here, of course, in general, Strunk and White, and that's even on the closer bookshelf in my office than how to write a scientific paper. So there, you can even just Google some of those things, and you'll get some hits that might be helpful.
Yeah, Strunk and White, Elements of Style, if you don't already own it, you need to. It's just everybody who writes anything needs to read it, and if you, it will improve your writing, and if you haven't read it in a while, read through it again, and it'll improve your writing again. It's, it's the best book ever written on the topic.
This How to Write a Paper, I remember reading that as a graduate student. It's very good.
But the best course I ever took was at Rockefeller. I think I mentioned this once before, long ago on one of our podcasts. We took 12 weeks of 2 hours each session on how to give a 10-minute paper. And it was a long, drawn-out procedure, which resulted in an absolutely lucid and understandable way of conversing in a 10-minute period of time, a single idea. What was the idea? How did you do it? And what does it mean? And if you can make a series of 10-minute papers and string them together into a grant, that's how you do it as far as I'm concerned, because that taught me how to be succinct and to the point. And that's the basic truth of writing a good grant, I think. And then, of course, you have to have an idea.
I think also it helps Ibrahim to pick some papers or propose or you want to write a thesis, right? Or maybe he wrote his thesis and he wants to write a proposal. But get some good ones and read them
exactly.
and learn from them.
Exactly.
You get a good paper that's succinct and puts everything forward, and just emulate it.
And the more you write, the better you will get at it.
Yeah.
And just to reiterate the outline, I live by outlines. It will absolutely save you when you're sitting there in front of a blank word processor. What am I going to do? And for stuff like grants and thesis and papers, and most of what I do is more freeform. But for things that are in science, there is always a formula. And so you start off with the format you're supposed to follow, and then you progressively work through it and start filling things in.
Yep.
Sometimes I do stuff that's like that, like a meeting report where it's structured according to the order in which the meeting occurred, and you can work your way through an enormous document just by breaking it down and saying, okay, I'm not working on this enormous document. I'm working on this paragraph right now. And it's just about this, and I already know what it's about, and I know what I have to say, and then you just say it.
Yep.
All right, let's do two more, okay?
Sure.
Mm-hmm.
Okay.
Alan, take Beverly's, please.
Okay. Beverly writes, I just listened to your interview with Dr. Lipkin, and I have a couple of questions and comments. I read the paper itself, but I don't understand the thing about the 9 patients and 9 controls that tested positive. This is about the XMRV story. I read the paper. I'm not sure if it was not fully explained or I just don't know enough to understand. I know for sure that my brain fog from CFS has been pretty bad lately. That's chronic fatigue syndrome. I got the impression that it was kind of waved off as some type of contamination or false positive. I noticed in the press conference that when Dr. Lipkin was asked about XMRV being a lab-created contaminant that was supposedly created by passaging prostate cancer tissue on a nude mouse, he dodged the question by discussing how scientists are working to come up with some guidelines on hybrid viruses that are being deliberately created. I'm sure that is very needed, but the question was about viruses that are being created accidentally. It seems to me there's an important story here about accidentally lab-created viruses and or natural hybrids and chimeras contaminating labs, reagents, and other products. Dr. Mikovits did talk about the amount of effort that the Rossetti lab went to in order to make sure their lab was clean. One incident in this soap opera sticks in my mind. One of the labs that got negative results admitted later that they had thrown out the results when a control had tested positive. Turns out that they used their lab personnel as controls, and they were sure they were negative because they got regular blood tests. That person may in fact have been exposed to XMRV and be positive, or it could just be more contamination. I think the lab contamination issue would make a good topic for a show. If you've already done one, please send me the link. I have an MPH in biostatistics, so I know a little about experimental design and things that can go wrong and how easy it is to come to false conclusions. So I enjoyed the discussion near the end with Dr. Lipkin about Koch's postulates and the respiratory disease study that went wrong. I'm eager to hear the program when his new paper on this comes out. And Beverly's in Austin, Texas.
The 9 patients and controls that tested positive, these are patients that had antibodies that reacted with, what was the antigen? I guess they're looking for XMRV antigens in the sera by an ELISA, and 9 controls and 9 patients were positive. You remember in this study, this was the multi-center blinded study that Lipkin coordinated.
Right.
None of the samples were positive by PCR, right?
Right.
But some of them were positive, both patients and controls, for antibodies against the virus. And he didn't know how to explain it, but there's clearly no virus present because the PCR is definitive. And the antibody tests can be funny. You know, they can be reacting with an antigen. And you were, he was saying that these people may have polyclonal activation where you make a lot of antibodies against different epitopes. And they react with many different antigens. So he doesn't have an answer, but he didn't wave it off. He just doesn't know the answer, and all we can say is it's not XMRV.
Right.
And, the idea of the thing about contamination is, I think we talked about it a lot in this XMRV study, right?
Yeah.
I mean, it was a year-long thing. I mean, I'm not sure what we would do for a show. We've also had an episode where we talked about, I think, a vector that contaminated some virus preps a long time ago. So I don't know what we would do for that. But
yeah, it's always a concern.
It's always a concern. But it's certainly if something comes up, we'll do it.
Yep.
Mm-hmm.
All right, the last one, Dickson.
It's Amy writes, Dear Doctors TWiV. That's an interesting way. I am a huge TWiV, TWIP, and TWIM fan, and was finally compelled to write after listening to TWiV 181, since I am currently reading the book The Family Who Couldn't Sleep by D.T. Max, a book about a family who has an inherited prion disease called fatal familial insomnia. Gee, I wonder if I have that. The author goes into the history of prion diseases and the discovery that they are not viruses or bacteria, but a protein. I'm about halfway through the book and I love it. I have also on my Nook the book The Wildlife of Our Bodies by Robert Dunn. He talks about what lives in or on us and is a big fan of Dickson's vertical farm idea. Really?
Don't read that book then.
Wow, I'm going to go pick up 3 copies of The Wildlife in Our Body. I have also had to say that my son likes to listen when we do errands in the car, and once commented that I listen to the most interesting podcasts. That's a great son you've got there.
Cool.
He really got a kick out of the viruses in video games episode. That was when Vince brought his son here. And also the zinc finger inspired by the NCIS episode. It's called Toxic, by the way. I am a year I am a year-old woman.
An ex-blank-year-old woman.
Oh, I am an ex-year-old woman who always has loved science since a small child, but did not have a chance to go to college. But I try to learn as much as possible on my own. That's fantastic. I've tried lots of science podcasts, and these are my favorite of all. Amy.
I think that's great.
Me too.
I like
Yeah.
that she
The
can learn
wildlife
science
of-
from us. So
Yeah.
thank you, Amy. Okay.
Or even is interested in science.
Well, that's the start, right?
I mean, that's counter to the most of the opinions of people that say, the uneducated majority of people that have no science background sort of poo-poo science. But this is not true at all. Here's a good idea. Here's a good person.
Well, a good show will get you interested.
There you go.
There you go. And that brings us to picks of the week, Dickson.
Yes.
Do you have a pick for us?
I don't. I actually don't. But I would like to recommend two websites that are run by NASA. One is
Those are picks of the week, aren't
Well,
they?
okay, we'll make them picks of the week. I mean, I'm a routine visitor to these websites, and I just love them. One is the Cassini-Huygens website for the mission, Cassini-Huygens, which is the Carolyn Porco monitored visit to Saturn, and all of the ramifications of that. It's just an amazing. There's a huge storm right now, in case you're interested in what the weather is like on Saturn. There's an enormous storm going on right now that you can go and watch. They've even got a video loop of it. That's quite fascinating. And the other one is there's a reconnaissance orbiter around Mars. Not the one that's crawling along on the surface, but rather the one that's going around about 200 or 300 miles above the surface with a super high resolution camera that captures these fabulous images of the surface of Mars. You can imagine yourself walking as the only person on the surface based on these amazing resolutions of photographs. So those, if you want to escape into another world that still exists.
Dickson.
I'm sorry, I can't help it. Those are my two picks, and I'll crawl off into a corner
I have to do so much editing.
and get better. I'm sorry.
I should let you edit it.
Yeah, that'll teach me.
I'll never get out.
That'll teach me.
Kathy, what do you have?
So my pick is both a press release video about a paper in Cell that came out in October and the paper itself. And the paper is entitled A Structural Basis for the Assembly and Functions of a Viral Polymer That Inactivates Multiple Tumor Suppressors. And it's from Ou et al. OU is the first author, and the senior author is Claudia O'Shea from the Salk Institute. And I did this paper for Journal Club today, and it's a really cool story. It's the first time a full-length adenovirus protein has been crystallized, and determining the structure of this has allowed for understanding how this very small protein assembles into a large random network array in the nucleus of the cell, partitioning off various things, the nucleosomes from the viral replication factories. And it's just got some cool movies, cool microscopic techniques. If we talk about it on a future TWiV, those of you who've already looked at it will be a couple steps ahead. It's just a really exciting story. I think it has implications for cell biology as well as virology. So,
Cool.
Wow.
Very cool. Yeah, we'll do this in the future. I don't know.
Yeah, and
when.
I kind of like these press release videos in lieu of or in addition to press releases. I'll probably change my tune on that in 2 or 3 years when I'm completely inundated with them, because I'm sure that'll happen. But for now, these are a nice novelty, and it's cool to see the researcher talking about the work.
right. It also is the sort of we had that same word in the reader emails, but it's the video flick for the paper
right,
itself.
Paper flick, yeah.
Paper flick, yeah.
Cool.
What are you looking at?
I'm just curious, as we've known about adenotol for a long, long time. How come this is the first protein that we've crystallized from this protein?
It's the first full length.
Why? Why?
Of the oncoproteins, I should have said that more carefully.
Okay,
Of the early proteins that get expressed.
Okay.
So it's the full length protein, and it was hard to do because polymer proteins are hard to crystallize because they aggregate.
Right.
So they had to make a mutant form of it and crystallize that. So very nice piece of work.
Nice.
Cool.
Good. Thank you. Alan, what do you have?
I have a really cool, I thought, Tree of Life illustration. So this is something called One Zoom, and they've put together this taxonomic tree that's very artistically drawn. What's cool about it is if you can explore a couple of branches of it just by clicking on the website. So, for example, if you go to mammals, you start out and you're looking at the whole mammal family tree, and it's just got very general categories. Okay, there are the marsupials, there are the elephants, and yada, yada. But then you can zoom. And you can zoom in all the way down to individual organisms and individual branches on the tree. And as you zoom in, more text appears on the individual, so you can look at the individual species of possum and see when they diverged from the main lineage and when they diverged from other marsupials.
That's brilliant.
Because you can keep going in and see nothing also.
Yes, you can. Yeah, eventually you'll go in and you'll see nothing but green.
Where are the viruses?
Well, I guess it's not up to date yet.
It's cool. I like that.
Zoom
nice
with
site.
a Z-O-O-M.
Yeah.
Z-O-O-M.
As in Zoom.
Cool. Thank you.
That's fantastic.
That is really cool.
Yeah.
I have a pick which is very unusual for me, and that's damn, my link is not working. Well,
That's a very typical link for you.
My link is not working. I like that link. I get that one a lot.
let's see if I can type it in. Yeah,
Oh,
it's
it
called
works.
Spot the Station. Yeah, it's not working on my Google Doc for some
Spot
reason.
the Station.
Spot the Station. It's a NASA site. And you sign up for alerts, and they tell you when the space station is flying overhead.
Right.
Cool.
So you can get a text or an email, and
Great.
you go outside, you look at it, and it moves across the sky because it goes pretty quickly.
Sure.
So I got an email yesterday that it would be over my house at 5:36 a.m. for just 3 minutes.
Huh.
You know, it's pretty bright, and you can see it. It's the third brightest object in the sky after the sun and the moon.
Cool.
Neat.
And this is
Neat.
a way to find it.
Yeah.
So that's pretty cool.
Yes.
I don't usually do astronomy things, but I thought
We're
that
all
was-
over NASA today, aren't we?
We
Yeah, we
are.
are.
We
And
are.
I actually I get emails because I want to. Well, 5:30, I wasn't out of bed yet, but I was
Right.
thinking about
By
it.
the way, apparently,
Yes, sir.
the latest rover on the surface has discovered something through its chemical analysis that NASA is waiting to make a big announcement about. And it's a mystery, and they don't
Yeah.
usually do things like
They
this.
found bacteria on Mars.
Well, we don't know. It's
I find
anybody's
this.
guess.
I have to say, I find this very, very annoying.
Yeah, I totally agree. It's to get you to listen to what they have to say. But
And that is just so ridiculous, especially coming from a government agency. You know, if
Yeah,
you have results, publish them.
Tell me
If
about
you
it.
Can't publish them yet,
Well-
then shut up.
I think they might want to confirm this before they say anything.
Well, then they shouldn't have said anything. That's the
Agreed.
problem,
No,
man.
Alan, I agree with you.
Arsenic, right?
Yeah, exactly. Arsenic life. And
Right. That's
They
right.
apparently have learned nothing. So.
Oh, well.
And,
Hey.
but then again, maybe it's going to turn out they're talking to Martians now, and I'm just going to have to be quiet.
They
but-
found remnants of life. That could be too. But guess what? We just had a meteorite hit the Earth recently, which came as a chunk of Mars, landed on Earth.
Oh yeah.
And we know it's from Mars because of the signatures of all the elements and stuff like that. So why can't chunks of Earth land on Mars?
Well, you know what this really indicates, though?
Yeah, it could.
It tells us that the Martians are now launching probes to us.
Yeah, that's right.
Bingo! I knew it.
All right. We have a couple of listener picks. One is from Ricardo, our friend in Portugal. He sends a story in so Nature has a website called Citable, a collaborative learning space for science, and there is a blog about science by students for students called Student Voices. He links to a story by Paige Brown called Once Upon a Time, The Possible Story of Viruses.
Ha.
So where they may have come from. So a nice little piece of writing here, which sort of ties in with the article we talked about today. It talks about Luca, the last universal cellular ancestor, not an Italian immunologist or an immunocompetent Italian.
Yes.
Right? And Dr. Forterre, who has been involved in identifying a lot of these big viruses. So that's a pretty nice piece of work there. And then we have Jim, our friend from Virginia, who recommends podcasts that might be of interest to our listeners, especially student The Encode podcast is almost an hour. Individual development plan you know about, but the 7-minute podcast will explain it to listeners. And then Experiment Reproduction. So we'll just give you the link to those. So thanks for that, Jim. And finally, Mark. Hey, TWiVome. Here is a way for listeners and hosts to inoculate against mathematics phobia. The USA's first Museum of Mathematics.
Yes.
It's opening in Manhattan on December 12th. 12/12/12. I wonder why they picked that date.
That's right.
Outreach to generate interest among youth is a key part of their activity. More information at momath.org.
Yep.
Ciao. Yeah, you want to go, Dickson?
Sure.
We could take an
Sure.
iPhone and record ourselves in there and then put it up on YouTube.
We could do that. We could do that. That wouldn't be wrong.
You're going to be in some foreign country on the 12th, aren't you?
No.
Okay.
Yes, I am actually. I'm going to be in New Jersey. Just remembered.
Foreign country. TWiV can be found on iTunes at twiv.tv and microbeworld.org/TWiV. If you like us and you want to help us out, the best way you can help us is to go over to iTunes and give us a rating on the show and leave a comment. That helps us to stay prominent, and that helps us to attract new listeners. We have lots of emails left. We're going to have to do another all email very soon before the end of the year. But if you want to send us a question or comment, we will get to it. TWiV at twiv.tv. Dickson Despommier. It can be found right here at Columbia University.
This is true.
Thank you for joining us, Dickson.
It's my pleasure as always.
Verticalfarm.com. And you know his other websites, too.
Right.
Thank you, Dickson.
You're welcome.
Kathy Spindler is at the University of Michigan. Thank you.
Thank you. I just checked to see when 12/12/12 is. It's on a Wednesday. And then I noticed that I already have marked on there 12:12 p.m. So I'm going to get an alert at that time. I have forgotten that I did that.
Huh.
I'm kind of a geek.
Cool. Yes, well, by definition.
So thanks for having me. It was fun.
Rich Condit never made it. He's getting his plumbing fixed, and he said he's sorry he can't make it this time.
Oh, well.
So another time.
First things
Maybe
first.
next Friday.
Yeah.
And Alan Dove is at alandove.com and on Twitter. Thank you, Alan.
Always a pleasure.
I'm Vincent Racaniello. Dixon's laughing.
No, because I love his response.
Because I always say always a pleasure.
You
well,
do
it is
always-
always a pleasure.
He
Alan-
said on TWiP this week, I said, thank you, Dickson. He said, always a pleasure. He said,
That's
I'm,
right,
I'm Alan Dove today.
No, you say it so well too, Alan, and I know you mean it. So I'm not
I do.
mocking you. I am acknowledging you as a warm and open and genuine person. Really,
Okay.
really,
On that note,
yeah.
I'm Vincent Racaniello, and you can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.